It is really a simple plug-and-play solution. It's been made to GMP grade. It has a drug master files. So it's simply just getting a bottle and then using it at the same concentration as you would currently use with DMSO at 5% or 10%. So most cells would just transit to DMSO to this solution without any problem.
Arctic fish survive in waters that would freeze most life solid. That biological insight, which translated into synthetic peptide chemistry, may be exactly what cell therapy manufacturing has been waiting for. In part 1, Steve O walked us through the DMSO problem in depth, the toxicity, the cellular damage, the regulatory pressure. Now we get to solutions. From cryopreservation performance data to manufacturing logistics to what a transition actually requires. This is where it gets practical. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Can you give us some examples, Steve, how this innovative approach helps cells survive better, maintain their viability, their function?
We have a presentation on various cell types, both on T cells and MSCs. So we have an example where fresh pan T cells isolated from donor apheresis were stored at 10 million cells per mil, and then they were frozen down for 40 days with XT Thrive solution. And post-recovery, they were able to proliferate as well as the fresh product. Another example was when we preserved T cells from 2 CDMO partners. The XT Thrive frozen product had almost equivalent or 80% secretion of IL-2, similar to fresh cells, whereas the DMSO containing product was around 60%. And in terms of the immunophenotype, they expressed CD4-8 markers. In terms of the functions of cytotoxicity, 4 competitors with DMSO were tested compared to the XT Thrive solution at the effector target ratio of 1:1 and 0.3:1. So XT Thrive had the best performance in terms of cell killing function over the other 3 DMSO containing products. In fact, one product had no killing efficacy at all at the lowest effector to target ratio. A third example is when CD4 and CD8 T cells were frozen for 7 days and then thawed, and nucleafaction of the CAR T gene was delivered. The efficacy was about 80% for XT Thrive versus 60% for CS10 cryopreservation solution. These are the T cell examples which we've seen much better than the CS10. In terms of the MSCs, we looked at holding the solution for 24 hours prior to freezing. We found that the viability of the cells that were held in XT Thrive maintained about 90%, but the DMSO dropped progressively in 4 hours to 85%, and then by overnight it was down to 60%. Then when we froze and thawed them, again, the recovery of viability was about 90% for XT Thrive and about 85% for CS10. And it continued to drop to about 60% over 8 hours, whereas XT Thrive was maintained around 90% in MSC cultures. And all these were done with both serum and serum-free conditions. And many products are moving into defined serum-free conditions, and cells tend to be more sensitive without the serum background. So we've seen this performance to be irrespective of serum-free conditions or serum. And then finally, when we put these cells back into culture in microcarriers, which is a scalable method for production of the cells, we saw immediate takeoff of the cultures post-thaw from XT-Thrive. But in DMSO, there was a 4-day lag and then marginal growth. So we had overall 2.5 times increase in the cell yield post-thaw.
To what extent, Steve, were these stark differences between XT-Thrive and DMSO due to ice crystal formation and differences, I'd say, in mechanism?
I think that is the key benefits of this. We have seen in microscopic studies that the ice crystals are relatively large in water, and both the antifreeze protein and the XT-Thrive product is between 50 to 10% of the size of the structure of the crystals in water. So most of the damage comes when you thaw, and these crystals just can cause damage to the membrane if the thawing is not rapid enough.
Let's shift gear here, Steve. So we have seen the stark differences between DMSO and this novel solution. We have also talked about the main challenges linked to DMSO. Now, another question we need to tackle is, okay, now we have a new product that seems to work well, but how does this affect established workflows? Washing, freezing and thawing, temperature. Can you give us a picture there? What changes there for the person doing the work in the lab?
So one of the first things we did was to do the whole time experiment over 24 hours. We learned in the early days that once you put the cells in DMSO, you have to process it quickly within 4 hours, but actually ideally between 30 minutes to 2 hours, which is fine when you're using small vials of cells that you can just aliquot 1 ml. But once you're processing, say 100 mils bags, or maybe you process a liter bag and then you aliquot to 100 ml bags, that workflow becomes very rushed. So that's why we did the whole time experiment over 24 hours to ensure that cells had to be processed over a full day. You want to see whether or not the cells would be affected. And sure enough, we found that the viability held over the 24 hours, which makes the processing much more convenient. And the back end also, because there was tests done where we injected the solution into animals at 100 times concentration that is being used for biopreservation, and there's no toxicity. There isn't the challenge to wash away the DMSO solution. And you could inject it as is. Post-thaw is like water. Well, not exactly, but it's non-toxic. You just inject it into the patient without having that extra wash step. So again, there's a reduced chance of contamination due to the manual wash and spins, and much simplified workflow from the point of view of point of care delivery.
How does this simplified workflow without an additional wash step after the thawing affect, or I'd say facilitate, distribution of these therapies to remote places? Because that's one of the challenges we are still facing in cell and gene therapy, that how are we bringing these therapies from the processing side, whether it's a hospital, whether it's a company, to the patient?
So we have tested the solutions in 4 degrees, minus 80, and minus 196 degrees. So in all these temperatures, the cell viability and performance are as good as or better than DMSO. So in the autologous therapy, you can hold them 4 degrees for as long as you need for transportation. And then in the manufacturing, you can process it in creating this master cell bag and a working cell bag at minus 80 to minus 196. And also for allogeneic therapy, you can handle much larger volumes and do long-term years of presentation and thaw them for localized therapy. So at all these temperatures, the solution is working well. One of the benefits of being able to work also at 4 degrees is that later on I'll tell you more about the solution where you can carry some of these cell types like organoids or organs for 3 to 5 days in cold temperatures. So it's never seen ice crystals. You just carry them for 3 to 5 days, transport across the country. Then this organs is still functioning after warming. So that again, it's the versatility of the DMSO.
So am I hearing correctly that this product can not only be used for single cells, but for organoids and potentially even in tissue engineering or some other areas?
Yeah, that's right. We don't have published data yet, but there is accumulated evidence that heart islets are functioning in this XT Thrive, and the XT Novo data for larger human organs will be coming out. So at ISCT May 2026 in Dublin, we should have more data on these different cell types, including organoids. And organoids have their own challenges because fairly large structures, about to 5 to 10 millimeters in size, and they need to be kept cold, or they need to be made fresh because there's no way to freeze them down or hold them in cold temperatures. And we think we have some good data to show for that.
Let's assume that my team and I have developed a T-cell process using DMSO, and now I'm listening to this podcast and I learned about this much better product to freeze and thaw cells. How easily can I switch over to this new product?
I think it is really a simple plug and play solution. It's been made to GMP grade. It has a drug master files. So it's simply just getting a bottle and then using it at the same concentration as you would currently use with DMSO at 5% or 10%. So most cells would just transit to DMSO to the solution without any problem. In terms of the manufacturing facilities, there's one in San Francisco and Vienna, and clients are welcome to visit them to audit the manufacturing process. We are also talking to some major distributors in the US and Japan to look at extending their reach globally.
Let's zoom out and look at the broader cell and gene therapy picture. There's a lot of innovation happening. DMSO is one of them. Beyond DMSO, what are some impactful areas you're seeing or some promising areas that you think are worthwhile highlighting?
So related to cryopreservation or beyond cryopreservation?
In general, beyond cryopreservation.
I think the biggest challenge with the stem cell-based therapies is long differentiation time from starting material to the final product. That's one. The secondly is maintaining the consistency and purity of the target population, whether it be, say, neural stem cells for the Parkinson's disease trials or cardiomyocytes for heart disease. So this is an ongoing challenge. So the third one is to perform this at a low cost of goods. Process optimization is key here, just as in biologic manufacturing, because they use so many combinations of growth factors and small molecules to get the final cell type over many, many weeks. I think the cost of goods challenge, because of the complexity of the process, is going to determine whether or not we have widespread use of these cell therapies.
What technology innovations have you seen that you think could solve one of these challenges?
I have seen some data from a company called Accelerated Bio, from a source of cells called trophoblast stem cells. And they had data showing that with a 1-day induction, they could make a cell type that secretes dopamine. And when injected into mice models of Parkinson's, they were able to recover the function. But that company has been having challenges raising funds to move into phase 1 clinical trial. So if some transformative technology like that takes 1 day to make a cell type that works similar to a dopaminergic neuron, that would be fantastic in terms of process, manufacturing, and cost of goods, but still not yet done in phase 1 clinical trials.
Such an innovation would move the needle forward, but it's a pity to see that disruptive technology stalls somehow because of a lack of funding. I mean, it looks very promising. Even later, perhaps you might run into some technical issues, who knows? But I mean, it's at least worthwhile pursuing the development and see whether it will work in a commercial setting.
If you had the opportunity, it might be good to get a venture capitalist or somebody on the other side who's able to give money and talk about what are the things to address.
Yeah, absolutely. And if one of the venture capitalists is listening, this is an opportunity. We are in the money game, yes, but I think we should also have a broader perspective. That's at least my personal view of the industry. And we should pursue technologies that don't have an immediate return on investment, because at the end of the day, we're in it to treat patients. So I think this is something, if I may say, in our industry, we could further develop also a bit more the social part of the development and not only the immediate ROI.
Absolutely.
Well, this has been great, Steve. Before we wrap up, what burning question haven't I asked that you are eager to share with our biotech community?
I think in terms of disruptive technology, XT Thrive, this single cryopreservation solution, will contribute significantly to cell-based therapies because it has zero toxicity. It preserves cells and tissues at a much better state than traditional DMSO for over 20 different cell types. Recovery of cells post-thaw, much more viable, functional, and healthier. Doesn't need a wash step, and it doesn't cause irritation or edema at the site of injection, which makes it easier to administer to patients. So I think this will be one of the key solutions in cell therapies that will be impacting both the current T-cell-based therapies and future stem cell therapies. Thank you for the opportunity to speak about this topic.
Absolutely. It's a pleasure. And if there is only one thing that you want our listeners to walk away with, what would it be?
If you're looking for a cryopreservation solution, just reach out and try Boho.
Okay. So this leads me to the final question, Steve. Where can people get a hold of you and a hold of this product?
So I'm on LinkedIn, and then my email, I don't know if you can share the text, is skwoh.so@gmail.com.
There you got it, Smart Biotech Scientist. You will find the link in the show notes. Reach out to Steve and his team. And thank you, Steve, so much for being on the show today and sharing about the challenges and also solutions in cryopreservation. Thank you so much.
Thanks, David. Cheers.
From Arctic antifreeze proteins to clinical grade cryopreservation, Steve O has shown us that the DMSO era may finally have a credible successor. The performance data is compelling, and the manufacturing implications are significant. If you're navigating these challenges in your own program, I hope this conversation gave you a clear path forward. And if it did, please take a moment to leave a review on Apple Podcasts or your favorite platform and share it with a colleague. Thank you for tuning in. I see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.